Hello, my name is Jason Flynn, a student in the Comparative Biomedical Sciences PhD program at North Carolina State University. Today, I will be giving an overview of sex determination in Drosophila, commonly known as fruit flies. In human sex determination, male or female development depends on the availability of a Y sex chromosome. The Y chromosome contains the SRY, or SARI gene, that stimulates male development. However, in Drosophila, sex determination does not depend on the presence of the Y chromosome. The Y chromosome does possess genes for sperm formation in adult flies, but sex determination relies on the ratio of X chromosomes to the number of autosome sets in a fly. This is referred to as the X to A ratio. If the X to A ratio equals 1, then the fly will develop a normal female. If the X to A ratio equals 0.5, then the fly will develop as a normal male. Ratios greater than 1 or less than 0.5 will cause flies to develop into metafemales and metamales respectively. These metasexes appear as normal male and female fruit flies but have lower viability. Ratios between 0.5 and 1 result in intersex fruit flies that contain portions of both male and female specific tissue. This is how Drosophila sex can be determined using chromosomal information, but what happens molecularly to result in different sexes? The central dogma of biology explains that DNA in the nucleus encodes genes. These genes are transcribed into pre-mRNA from DNA. Before leaving the nucleus, pre-mRNA is processed. This includes addition of a 5' cap and a poly-A tail, and RNA splicing. Splicing of the pre-mRNA transcript allows for a single gene to produce proteins of different composition and length. After processing, the mRNA is exported to the cytoplasm and translated by ribosomes to produce the protein. First, let's understand key genes involved in Drosophila sex determination. Sex-lethal, abbreviated SXL, is a gene that encodes a splicing repressor. Sex-lethal promotes skipping of normal splice sites by interrupting factors such as SPF45 and U2AF that normally promote splicing. Transformer, abbreviated TRA, is a splicing activator that enhances splicing at its bound position in the transcript. Finally, double sex, abbreviated as DSX, is a transcriptional repressor that dictates which genes are turned off in males versus females. Ultimately, the splicing effects of sex-lethal and transformer impact which exons are included in the final double sex transcript. In a normal male fruit fly, an optional exon in both the sex-lethal and transformer transcripts is retained during processing. This optional exon in both transcripts contains an early stop codon that leads to production of shortened, non-functional proteins. Without a functional sex-lethal protein, the optional exon is included in the transformer transcript and a non-functional transformer protein is produced. In the absence of a functional transformer protein, a long double-sex mRNA composed of exons 1, 2, 3, 5, and 6 is produced. This long double-sex transcript is translated into a male-specific double-sex transcription factor that suppresses female development genes and promotes fly maleness. In a normal female fruit fly, the optional exon in both the sex-lethal and transformer transcripts is spliced out. This results in functional sex-lethal and transformer proteins. The functional sex-lethal protein acts as a splicing repressor. In its own transcripts, sex-lethal interacts with SPF45 to block a splice site, promoting the skipping of the stop exon. This aids in autoregulation of the sex-lethal gene. Sex-lethal protein also promotes skipping of the stop exon in the transformer transcript by blocking U2AF binding and forcing it to bind at a different splice site. This allows for the production of a functional transformer protein. As a splicing activator, transformer works with other proteins to differentially splice the double sex transcript compared to what is seen in male flies. Splicing after transformer binding results in double sex transcript composed of exons 1, 2, 3, and 4. This shorter double sex transcript is translated into a female-specific transcription factor that suppresses male development and promotes fly femaleness. Thank you for watching this overview of sex determination in Drosophila.